cute little champions, I welcome you all to score 360 out of 360 in NEET Bio within 250 days. And today we are going to talk about two very important vectors and competent host. So about pBR322, even though we studied it earlier in the previous session, I thought I need a little bit of improvisation there. So I have redone it, and I think this time you might understand a bit better. And also when you relate with this thing, the other vector, it becomes all the more clear. Okay, so let's begin. And yeah, what all did we cover in our previous session? We spoke about the types of the cloning vector. So for plants you have agrobacterium tumefaciens, for bacteria, okay, host you have different thing, for human beings you have retrovirus and things like that. Okay, now features of a cloning vectors, about ORI site, about what all things they should be having, for example selectable marker, the cloning site and all these things. Then we spoke a bit about back and yak, that is bacterial artificial chromosome and yeast artificial chromosome, a bit about it, and I also told you what type of questions could come. The homework that I gave was also something related to that, so we will see it. And then we spoke about pBR322, about which we are going to study a bit more properly this time. Okay, now the number of questions from this chapter was 5 the previous year. We can expect a lot more, or at least 5 or 4 would be there for sure. And this is a chapter which could help you score really well, because it does not take too much of your time as well. Okay, it's not a too much time taking chapter, and it's interesting, something different from what we study. It's not monotonous, so you should be definitely trying it out. Okay, now going ahead, the homework that I gave you, let's see who all got it correct. So, commonly used vectors for human genome sequencing are, so commonly used vectors for this thing. So, we studied that in the case of human genome project which was done, it was said that we need a vector that can accommodate a larger gene of interest. Okay, we need a vector which can accommodate a larger gene of interest. So usually the plasmids and everything that you see, they have a shorter gene of interest sequence. So for example, this is a plasmid, you can put short gene of interest sequences over here. Okay, but when you talk about these guys, that is back and yak, bacterial artificial chromosome and yeast artificial chromosome, this is put in the case of bacteria, this is put in the case of yeast. Now what do they do, how they are different? Basically this chromosome, no, BAC and yak, they can accommodate a larger gene of interest, and that is what we need in the case of human genome sequencing. So basically what was the correct answer? It was back and yak. This came in 2014, my dear children. So if you pay attention to all these little details, you will be scoring really well. Okay, now going ahead, let's talk about pBR322 vector. So here is a picture that is given from your NCERT. Okay, here is the picture that is given in your NCERT, and I am just going to explain a bit about it. Okay, so a little bit we have already studied, so I won't be taking too much of your time, hopefully. So the first thing that you have to remember is that it has 2 genes for resistance against drugs. There are 2 drugs which is and it is resistant to it. Okay, it is what, children? It is resistant to it. That means if I am putting this drug where this particular guy is growing, it will still grow because it is resistant. It has the ability, the power to resist any sort of change. Okay, just give me a second. Okay, this gene is for tetracycline resistance. It's a drug, it is known as tetracycline resistant. Okay, it's a drug that is known as tetracycline, and this particular gene is tetracycline resistant gene. Over here, this is another gene which helps this particular organism to be resistant against something that is known as ampicillin. Okay, this is again a drug. So we have 2 drugs, and this particular plasmid is kind of resistant to it because it has the resistant gene. Okay, now you might be already knowing that basically if it's a plasmid, it will be definitely having an ORI site. So there is an ORI site. Okay, ORI site is where the DNA polymerase will come and attach and do the replication and everything over there. But it needs certain proteins also for replication, and that proteins are coded by ROP. It's another gene that will be coding for the proteins required for replication and everything. Cool. Now if it's a plasmid, it should be having sites for enzymes to act, because enzymes will come and act on it, cut it, and then you can put the gene of interest. So you need sites, recognition sites for different enzymes, right? So over here we'll start with tetracycline. Over here, this patch, this gene, which is offering resistance to tetracycline, it also has recognition sites. It also has sites for one very important enzyme, that is BAMH1. Okay, what do we have, children? BAMH1. Along with that, there is SALH1. There is a recognition site for SAL1, not SALH1, it is SAL1. Cool. So, 2 enzyme recognition sites are also over here. For example, if I put this enzyme, it will go and make a cut over here in the tetracycline resistant gene. Now, will it be active? No, it would not be active. So, anything can happen. I'm just talking about the recognition sites, what all are there in this particular plasmid. Okay, going ahead, what do we have over here? In the case of ampicillin resistant gene, here also you have 2 sites. One is for PVU1, okay, another is for PST1, okay, so here also you have 2, here also you have 2. Now, you saw PVU1 over here, okay, there's an enzyme whose recognition site is over here. You have one more enzyme, that is PVU2. Its recognition site is over here, and I'll mark it in different color. Ma'am, is the structure very important? Yes, children, structure is important. You have to remember it. Okay. Now, going ahead, there are recognition sites for 3 very important enzymes over here. Okay. Starting with EcoR1, that's the major guy. So, we have EcoR1. Okay. Then we have CLA1. What is it, children? CLA1. And then we have hint 3 or 2? 3. Yes, it was 3. So, how the question comes is that they will say that we are going to use PVU enzyme for this recombinant DNA technology. Over here we are going to use this particular restriction endonuclease. Will it bring out any change? Yes. If you are using this particular enzyme, it will make a cut over here. Now, if it makes a cut over here and if you put the gene of interest over here, do you think that it will still show ampicillin resistance? No, it won't show because it is hampered. So, if you are not aware about the structure, will you be able to solve the question, student? You won't be able to solve the question. Okay. So, the first, this is the structure. Okay, you call this PBR. It is Bovine Rodriguez, that is why B and R stands for, and it's 322, the number of the plasmid. Okay. Now, the thing that I am going to do is, okay, I am going to make use of one of this enzyme and make a cut over here and put my gene of interest over here instead of tetracycline resistance gene. So, now the situation is kind of different. Over here, I will be still having my ampicillin resistant gene. Okay, but I would not be having my tetracycline resistant gene. Instead of that, what will I have, children? I will have my gene of interest. This is what? This is the gene of interest. Okay, and I am writing this is ampicillin resistant, but it is not tetracycline resistant. Okay, that means if you put ampicillin, it will still survive because it has a resistant gene. But if you are putting tetracycline, it won't be able to survive. That is the thing. Now, what are we going to do with it? We are going to experiment. We are going to bring out this change and let's see who all got this particular plasmid. Now, we are going to treat it with bacteria and everything, the host. Okay, and we will see which all host got it and how we can deduce whether it got it or not. That is a complete scenario over here. So, where shall I make it? Shall I make it here? Chalo. Let's make it here. So, over here, I have my recombinant DNA with ampicillin resistant gene. Okay, and then I have the gene of interest. This is what? This is the gene of interest and this is ampicillin resistant gene. Okay, cool. Now, I get 3 different scenarios, my dear children. One is that in the host, this particular plasmid has not gone. Okay, in this particular host, this particular plasmid has not gone. So I'll write no transformation took place. Okay, second scenario is where I have this. Okay, this is also a host, and in this host I got my plasmid, okay, which had the ampicillin resistant gene and my gene of interest. Now this one you call it recombinant, you call this as recombinant plasmid, my dear children. Now there could be a plasmid in which the gene of interest has not come because of some reason. The gene of interest couldn't make itself up there. Okay, so over here you will be still having the tetracycline resistant gene. Over here you will be still having the tetracycline resistant gene and the ampicillin resistant gene. So the second scenario is when the host is taking a non-recombinant DNA. It is taking a non-recombinant DNA. So over here, or the plasmid. So over here I have the ampicillin resistant one and also the tetracycline resistant one. Okay, now I have three different hosts. I want to check which one got the transformant, or in which one transformation has taken place. Transformation means basically I am passing down my DNA. That is what you mean by transformation. Okay, I want to check which one has transformation taken place over here. In which one has transformation taken place? So that is my question. So for that, what do I do, children? I know some of them might not have tetracycline resistant gene, so I cannot do the test with tetracycline. Instead of that, what do? Is I can grow them in a medium that contains ampicillin. Now this guy which does not have the plasmid that contains ampicillin resistant gene, will it be able to survive, children? No. So this guy is out of the race. This guy is out of the race. So I can say that this is a non-transformant. What can I say? That this is a non-transformant, my dear children. This is non-transformant. What about this, children? Again, this is a transformant. This is a transformant. That means it has got the plasmid, even though it didn't get the recombinant plasmid, it still has got the plasmid. So I say that it is transformed. The DNA has come from outside, it is transformed for sure. I do not know whether it has a gene of interest or not. That's the only concern. So this again, it will be able to survive since it has the ampicillin resistant gene. Here also it will be able to survive. So I call both of them as transformants. Okay, so in the first round, I treated with ampicillin. I got to understand that two of them, they are transformants, whereas one is not. So I just exclude this out. Now I take these two hosts, okay, and I would want to check which one of them has the gene of interest. How can I do that, children? For doing that, so this is a scenario where this is a host, okay, and it has this. So over here, this is the ampicillin resistant gene and this is the gene of interest. Okay, then there is a host which has the plasmid that has both ampicillin resistant gene and also it has tetracycline resistant gene. Okay, so I am writing A resistant and R resistant. Over here is just A resistant and this is gene of interest. So if I treat it with, now if I want to test which one has the gene of interest and if I treat it with tetracycline, suppose if I treat both of them with tetracycline, then the one that has gene of interest would not be able to survive because there is no resistant gene for tetracycline. So all of them will get killed. So I don't want that. I just want to know which one has the gene of interest. That's the concern that I am having. So if I treat with tetracycline, all of them will get killed. This won't be getting killed, okay, because it has the gene for resistance. Right. So what can I do? Instead of treating it with tetracycline, what can I do, children? I can make a replica plate. Okay, I can make a replica plate where I am using a sheet to transfer some part of it, okay, some bacterial colony over to the next plate. Okay, and over here, this is a replica plating. Okay, I am putting something over here and then I'm putting it over here, a blot, okay, such that some of it could get transferred over here, and I know that these are the guys that I'm looking forward for, and these are the guys that does not have or basically has the gene of interest or not, okay. So exact copies are made over here, that is why I call it replica plating. Now, over here, I will do what I was planning to do there. I will now treat it with tetracycline. Children, I will now treat it with tetracycline. So what will happen? If this is a scenario and there is another scenario like this, okay, where I just have ampicillin resistant gene and then there's my gene of interest. And now if I treat the replica thing with tetracycline, what will happen, children? Will this survive? Yes, this colony will be surviving. This colony will definitely be surviving because it has the tetracycline resistant gene, and this colony does not have the tetracycline resistant gene because it has the gene of interest. So this won't be able to survive, and that's how I will get to know that basically I made a replica plating. That's how I'll get to know that this guy from where I took, they have the gene of interest, and now I can take them and make their multiple copies. Now it's totally fine, okay. So here it has the recombinant plasmid. They are recombinants, children. What do I call them? They are recombinants, whereas they are not recombinants. They do not have the gene of interest, nothing. Both of them are transformed. Both of them, if I see, they are transformed. So these are transformants for sure. These are transformants for sure. But are they recombinant, children? Only this one is recombinant. Only this one is recombinant, whereas this is non-recombinant. You get it? So how transformation is different from recombination, that is something different. If I have the gene of interest, then I call it recombinant plasmid. If I do not have, I cannot call it recombinant plasmid. And this is how things work. Okay. And these things, these enzymes that you see over here, the genes basically, you call them selectable markers because of them I am able to select which one has gene of interest and which of them does not have. If I do not have this particular sequence at all, then how can I understand it? So they become selectable markers. I am able to select them. Okay. So basically your restriction enzymes, okay, genes, basically the genes of the restriction enzymes are acting as selectable markers, my dear children. This is a very important point, keep in mind. Okay, so that is about pBR322. Fine. Now that is about pBR322. I have told you there is one more vector given in your NCERT. The name is not given, but they explain the entire scenario. So we have to study about that also. That is pUC8 vector. What about them, children? pUC8 vector, this is another plasmid. Okay, this is another plasmid which also has ampicillin resistance gene. And here there is another gene which you call as LAC-Z gene. Remember LAC-Z gene? It was responsible for making beta galactosidase. This is an enzyme that it codes for. So LAC-Z gene usually codes for beta galactosidase. Okay. Now I'll tell you the scenario. If beta galactosidase comes in contact with X-gal, which is another chromogenic substance, chromogenic as in it will show up its color if it is interacting with something like this. Okay. Then it will show up the color, it will change its color. Okay, there is some breakdown happening, which is why the color is being emitted. So when beta galactosidase comes in contact with this guy, that is X-gal, that is a chromogenic substance, it gives a blue color. It gives a blue color. Okay, that is a thing that you should be knowing. So over here, in this case, this is a normal scenario that happens. So every time if I am putting in this particular medium, if I am putting X-gal, what will happen? This will give me a colony of bacteria that has this particular thing. Okay, the blue color colony will be happening, okay, or being made over there. Now if I do a bit of change over here, and in this particular plasmid, if I alter it, so over here the ampicillin resistant gene, I'm just keeping it like that only, but here instead of LAC-Z gene I am putting my gene of interest. Instead of LAC-Z gene I am putting my gene of interest. So obviously beta galactosidase activity won't be there, because obviously that enzyme is not being produced because I have altered the LAG-Z gene, right? So no beta galactosidase. Now I have two scenarios. This is the first scenario, this is the second scenario. I have two colonies, okay, and then I put it in a plate. I treat it with ampicillin. I get to know, okay, first thing, basically I have to put it in a host. First thing, I have to put it in a host. Same thing happens again, okay, same thing happens again. Just give me a second. Where can I draw it? This is a huge problem. Just give me a second. Okay, wait. Yeah, I have added one more slide. Okay, so I have this scenario, children, and now I'm going to show you what happens next. Where is it? Okay. This and this with ampicillin resistant gene over here, and then the LAG-Z gene over here. Okay, this is the non-recombinant. This is what, children? The non-recombinant. Fine. Now there is one more. There is one more, that is recombinant. So in them, what will happen, children? I have the ampicillin resistant gene over here. Okay, and then I have my gene of interest. So this becomes what? This becomes recombinant. So again, when I put it in the host, I have three different scenarios. One scenario will be like this, where this guy is there, okay, where I have this guy. So I'm not entirely writing. I have the ampicillin resistant gene and I also have the LAG-Z gene. Okay, so this is the recombinant, one thing. Second thing that I can have is this, sorry, this is non-recombinant, okay. Second thing is like this, where I have the recombinant, that is ampicillin resistant gene is there, and instead of the LAG-Z gene, I have my, what is it? I have my gene of interest. Okay, now the third scenario, again, where the transformation, the transformation itself has not taken place. No transformation. Okay, so what do I do, children? First thing that I can do is to understand which one is transformed and which one is not. For that, what will I treat it with? All of them, if you see, both of them here, they are ampicillin resistant. So the one that will be having ampicillin, the one that will be having the plasmid, okay, one host that will be having the plasmid will be showing what, children? Ampicillin resistance. So I can put it first in the medium, I can put ampicillin, and I will see that both of them, they will be surviving. So both of them, they are, children, transformed. Both of them, they are, children, transformed. Okay, now over here transformation has not taken place, so this guy goes out. So this guy goes out. Now I make a plate, I put both the host over here. Okay, I put both of the host over here, and now I'm going to treat it with X-gal. I'm going to treat it with X-gal, my dear children. Now what will happen? There will be a colony of bacteria. There will be a colony of bacteria which would be giving blue color. Okay, and there will be a colony of bacteria where there won't be any color. So this will be a colorless colony, whereas this colony will be in blue color. Why is it colorless, children? Because over here I do not have LAG-Z gene. If LAG-Z gene is not there, will I have beta galactosidase? No. If beta galactosidase is not there, will it go and react with X-gal to produce the blue color? No, children. But over here I have the LAG-Z gene. LAG-Z gene is there, that means beta galactosidase is there. Beta galactosidase is this, and this reacts to give the blue color. So I have a blue color colony and have a colony of bacteria that does not have any color. So I will choose them. What will I choose, children? I will choose them. This is the gene of interest, and this is how the question comes. Will you choose the colorless one or will you choose the colorful ones, the blue color ones? Read the question carefully and then only go ahead. The scenarios could be changed, but read the question carefully and then only go ahead. This is the story of pUC8. This is the story of what, children? This is the story of, I made P bigger. This is the story of pUC8. Okay, now one very important thing that I want to explain with this is that this and the previous one, that is this, okay, when we are studying about pBR322, you notice one thing. You had the tetracycline resistant gene in that you made a cut and you put something else. So over here, am I not inactivating this particular gene? Am I not inactivating this particular gene? Yes. How am I inactivating? I am inserting my gene of interest, which is why it gets inactivated. So I say that this procedure is known as insertional inactivation. What do I call it, children? I call it insertional inactivation. I am inserting my gene of interest, which is why this particular gene is getting inactivated. Here, similarly, in the LAG-Z gene also, I am putting my gene of interest over here, which is why I am inactivating this guy over here. So this is known as insertional inactivation, which is given in your textbook, and you have to know about it for sure. Okay, now let's have a quick read of your NCERT from this particular portion, that is pUC8. About pBR322, we studied it earlier, so I'm not going ahead, but I'm telling you for pBR322, please know the names of these guys. Remember, everyone has 1111 except these two guys, PVU2 and HIND3. Rest everyone is 111111. So BAM, SAL, PVU, PST, PVU, EcoR, CLA and then HIND. Okay, either way, however you can try to remember it, you can use your own mnemonics for that. Okay, and if you have come across some mnemonic like that which would help you remember, please put down in the comment section so other children also can make use of it and they can also remember, right? It's a very good deed to help, no? Yes, chalo. Now, about pUC8, I told you this term is not given in your NCERT, but about which it has, everything is there. So we should be knowing about it. So selection of recombinants due to inactivation of antibiotics is a cumbersome procedure because it requires simultaneous plating on two plates having different antibiotics. Therefore, alternative selectable markers have been developed which differentiate recombinants from non-recombinants on the basis of their ability to produce color in the presence of chromogenic substance. So using too much amount of antibiotic resistant gene and stuff like that, it's obviously a very difficult process. You have to use two different plates and you have to do the test and everything over there. So that becomes difficult. So instead of that, why not use something which makes the use of the chromogenic substrates, which has some color change and everything. It's a lot more exciting and a lot less time taking as well. Okay, just give me a second, guys. Yeah. Now, in this, a recombinant DNA is inserted within the coding sequence of an enzyme. Within the coding sequence of what, children? An enzyme, beta-galactosidase. This results into inactivation of the gene for synthesis of this enzyme, which is referred to as insertional inactivation. What was this gene, children? The name of the gene is LAG-Z gene. Okay. Now, the presence of chromogenic substrate gives blue-colored colonies if the plasmid in the bacteria does not have an insert. So, if it does not have the insert, obviously it will be giving a blue color, okay, because beta-galactosidase is there and I am putting my chromogenic substance, which is X-gal. Now, presence of insert results into insertional inactivation of beta-galactosidase gene and the colonies do not produce any color. These are identified as the recombinant colonies. That question always comes. Which one of them will be identified as recombinant ones, children? These are the recombinant ones, the colorless ones. Please do not say the blue color ones are the recombinant ones, okay? These are the recombinant ones. So, transformed, yes, both of them are transformed, but if you talk about recombination, this one has a recombination, it does not have. Cool? Works? Will you be able to remember these two very important vectors? So, around which questions have come, so I want you to practice it as well. Okay, few of them I am only making you practice things, but still. Now, comes the story of competent host. So, over here in your NCERT, there was too much of confusion regarding competent host. What do you call, when do you call a host as competent? So, a competent host refers to a cell that has been prepared to efficiently take up foreign DNA such as plasmid during a process known as transformation. So, you know, in a process that is known as transformation, if this is a bacteria, I want to put my plasmid into this particular bacteria. My plasmid as in the gene of, as in, not Jasmine, okay, as in the gene that I want to put it over here, okay. So, basically, this process is known as transformation, but it's not a very easy process, okay. It's not very easy process to make the DNA enter into the cell and inside the nucleus and all these things, okay. In the bacteria, they do not have nucleus, in the cytoplasm. So, whatever is this. Now, this process of making a particular host capable of undergoing transformation is known as competency. Okay, what do you call it? It becomes a competent host. When I say that I am okay, I am perfectly okay for transformation to take place inside my body, that means my body has underwent certain changes, which is why I can take the DNA up very easily during the process of transformation. If for suppose I say there is no change in my body, I cannot allow the entry of any particular DNA inside my body or inside my cell, that means basically I'm not a competent host. I'm not a competent host. A competent host is a one that has been prepared for taking up the foreign DNA. Now, the question is, why can't the host naturally take up the DNA? Why do you think that it cannot easily get inside, the DNA can easily get inside and do all the things? If I mix it with the DNA and I suppose I had the DNA over here, okay, mixed in this particular substrate, and then I have my E. coli, then why doesn't it naturally take this up? Okay, or why is it very much difficult for making the DNA enter the host, even if it's not naturally possible, still, why is it too much difficult? The answer to this question is that DNA, if you know, it's negatively charged particle. What is it, children? It is negatively charged. Now, if we talk about our lipid bilayer, okay, you know there is lipid bilayer, right? Lipid bilayer, as in there are two layers. One is this, and the other one is the one that I am making over here. Okay, so this head region, if we see, it is hydrophilic. It likes to be in water. Okay, and then there is leg region, the tail region, you say. Suppose they are having shoes. They are like, we don't want water, we hate water. So this is hydrophobic. Okay, it does not like water at all. Now the problem is, if something wants to get passed down, so obviously this should not be having any water, nothing should be there if it wants to get passed from here, because you know the inner region is hydrophobic. That is why they are putting the leg inside and they are staying there. Now the problem with the DNA is that DNA is negatively charged. That's the one big problem. Okay, and over here the top region that you see over here, it is also negatively charged. So negative comes, it will repel. Obviously negative, for example, there is a DNA, okay, it comes, it will repel. It does not want it to get inside. That is the first problem. And then the negative charges that you see attract water molecules. Okay, what does the negative charge do, children? It attracts the water molecules, making the DNA highly water soluble, hydrophilic. Okay, so now the DNA, which is negatively charged, it is also wanting, okay, it is also going to attract lots of water molecule, which makes it hydrophilic. Now, do you think a hydrophilic thing can pass through an hydrophobic thing? No, it becomes a lot more difficult. So the hydrophilic and the negatively charged DNA is naturally repelled by the hydrophobic core of the lipid bilayer in the cell membrane. So initial problem was the hydrophilic head was there. Okay, this is negatively charged, and you are putting the DNA that is also negatively charged. So obviously it will repel. Now forget about it. The next problem that comes is that you have a hydrophobic core, and your negatively charged DNA, if it tries to get inside, it is hydrophilic in nature. It will try to attract more water molecules. So can it easily pass through this particular core? No, it becomes difficult, which is why I say transformation is not an easy process. Over here, you saw that one of them was not getting transformed, and this is totally fine. This happens usually because of our mechanisms to not allow any entry of foreign particles. Okay, that is a major thing. So over here also it becomes a lot more difficult. So ma'am, what is the option that we are having? The option that we are having is different for different organisms. Okay, so usually bacterial cell and everything, they undergo a mechanism that is known as heat shock. Okay, what do you call it? Heat shock. So for that, what is done? Let's see that. So this is done by treating them with a specific concentration of a divalent cation. Divalent as in it has two valency. Okay, so usually calcium and everything is used, calcium, magnesium and things are used. So over here, what I am doing, children, I am going to treat my particular host with divalent cation. What is the use of divalent cation? Basically, the thing is that the negative charges, no, it gets shielded. The cations that are there, okay, it will shield the negative effect, like how you shield with a blanket, right? Similarly, it will shield with the cations which are produced, which are being given, okay, such that the negative and negative comes together. When it comes together, it won't repel each other. So you are shielding the DNA as well as this particular host lipid membrane with the help of the divalent cations. Okay. So in your NCERT, it is not given entirely why do you use divalent cations. I am telling you it's for shielding effect. It's for shielding effect. Don't go very much inside, very much deeper. Just understand that you do not want negative to come and repel, which is why you are providing a shielding effect to both DNA and the lipid bilayer. Fine. Now, so over here, it is treated with a concentration of divalent cations such as calcium, which increases the efficiency with which DNA enters the bacterium through the pores in its cell wall. Now, recombinant DNA can then be forced into such cells by incubating the cells with recombinant DNA on ice. So, first thing that I am putting is, there will be my plasmid of interest or my DNA of interest, and then I am treating the whole thing. Okay, what is happening, children? There is a recombinant DNA that I have in a plasmid, suppose. Okay, and now I am incubating the cells with the recombinant DNA, and the temperature is quite low because it is icy. Okay, now what I am doing is that instantly I am changing the things. Okay, instantly I am going to change the things over here. I am going to give it a heat shock. Heat shock as in the degree will be 42 degrees Celsius. The temperature is 42 degrees Celsius. So initially it was cold. Now I am giving it heat shock. What will happen, children? Basically there will be certain transient pores. There will be certain transient pores. Transient pores as in for shorter time there will be pores. For shorter time there will be pores, children. Okay, so the bacteria is like, what is happening to me? So initially it was closed, everything was closed. When heat shock is given, it creates transient pores. By this time the DNA enters. So now the plasmid with the recombinant DNA is over here. Okay, now what I am going to do is that again I am going to treat it with children, ice. So what will happen? All the transient pores that you see over here, it will all get closed. So initially what was I doing? I was treating it with, children, a specific concentration of divalent cations. Okay, fine. And then I am putting my recombinant plasmid as well as my host cell in ice. Okay, then I was providing heat shock treatment, that is almost 42 degrees Celsius was being provided. At this point of time, what will happen, children? There will be transient pores being made over here. Small little pores are made over here. And now what will happen, children? By this time, with the help of transient pores, whatever recombinant DNA is there, it will get inside. Okay, it will get inside. And then what is happening? I am again treating with ice. I am again treating it with ice. So all the transient pores, they will close up, and now I have the recombinant plasmid inside. Okay, so initially that is what is written over here also. Recombinant DNA can then be forced into such cells by incubating the cells with recombinant DNA on ice, followed by placing them briefly at 42 degrees Celsius heat shock and then putting them back on ice. This enables the bacteria to take up the recombinant DNA. So this is how the things are working in the case of bacteria. Fine. Now things are different for other organisms. For example, if we talk about plants. Okay, if we talk about plants, they have something that is known as gene gun. Okay, animal cells, I say they are not really strong. Okay, so they cannot undertake gene gun. Instead of that, they have another mechanism of transforming things, that is micro injection. In the case of plants, you know, they have an extra layer that is known as cell wall, which makes it a lot more difficult, right? So what is done is that the gene of interest is put in a medium, and then a gun-like thing is being provided such that it makes, with the force that it is being put over there, not the gun that you are thinking of, okay? With the force that is put over there, what happens is that this particular gene of interest, or basically the recombinant DNA, gets inside the nucleus or the cytoplasm, wherever it is needed, where it is not put in the cytoplasm because you know there are so many enzymes, nucleases that will cleave the DNA. So we do not want that. We want it to safely reach the nucleus, and it is made to reach the nucleus with the help of gene gun. Okay, and the next thing over here is micro injection, where again in the case of an animal cell, what happens is that you put this competent host, or basically the host, under the microscope, and you have a micro pipette sort of thing. You have a small pipette sort of thing, and this pipette will be having your recombinant DNA. This pipette under invigilance is put inside this particular nucleus. This is known as micro injection. This is done in the case of animals. Now how this question comes, children? Which one is used for animals? Which one is used for plants? So gene gun, because you see plants, they are very strong. They have the cell wall and everything. So they can undergo an attack by the gun. So gene gun is used for that. But as we are very sensitive, so we need micro injections only. Okay, so that is what exactly is written over here. But apart from that, gene gun, what is it made up of and things like that is also written. So let's read it. Okay, so in a method known as microinjection, recombinant DNA is directly injected into the nucleus of what children? Animal cell. What happens if you put it outside children? There will be another enzymes which are nucleases, which would cleave the DNA. Which could cleave the DNA. Now, in another method suitable for plants, cells are bombarded with high velocity microparticles of gold or tungsten coated with the DNA. So, what happens is that you have gold or tungsten. Please remember both the terms. Okay, you have gold or you have tungsten, and what do? You coat it with. You coat this with the DNA. Okay, and then so with high velocity microparticles of gold or tungsten coated with DNA, it has been bombarded inside the nucleus such that everything is reached there. Now, this is known as gene gun, and it is also known as biolistics. Okay, biolistics. So, if the question comes that biolistic is used in the case of plants, is true or false? Basically, it's true. The gene gun is also said as biolistics. Please do not forget. So, for that, what are we using, children? We are using high velocity microparticles of what? Gold and tungsten, which is coated with DNA, and then we are inserting it like that. This is also known as biolistics, or it's also known as gene gun. Okay, now we have animal. For animals we have microinjection, and the last method uses, okay, so apart from both of them, so we studied once about bacteria. It uses the heat shock treatment for getting things done. Okay, then you have plant, then you have animals, and lastly it is saying that there is one more method, basically where you are using the disarmed pathogen vectors, which when allowed to infect the cell, transfer the recombinant DNA into the host. Disarmed as in, for example, there is a gene that could cause the trouble. Okay, I am removing this gene, I am putting my gene of interest. So, I am disarming this particular pathogen. What do you mean by disarming? Basically, I am not cutting its arm off, it does not have an arm. Basically, I am making it helpless. It's a very cruel way of doing things, but yes. Okay, so basically the power that it had, I just took that away, and I am putting my gene of interest over there, which makes it disarmed. That now it won't be able to infect me. It's not pathogenic anymore. Got it? So this is the transformation through microinjection and gene gun. Works? Now, okay, just give me a second. Biolistics or gene gun is suitable for what? Disarming pathogen vectors, transformation of plant cells, constructing recombinant DNA by joining it with vectors, or DNA fingerprinting. This came in MEMS 2012. Tell me the answers in the comment section. I will be discussing the answers later on. Okay, once I have given you all the questions, try to solve it. It's very much important. If you're not solving it, if you're just relying on me, that will be trouble. That's why. Now, for transformation, microparticles coated with DNA to be bombarded with gene gun are made up of what? 2012 question. Silver or platinum, platinum or zinc, silicon or platinum, and gold or tungsten. Okay, this is the second question. Third question. In genetic engineering, the antibiotics are used to what? As selectable markers, to select healthy vectors, as sequences where replication starts, to keep the cultures free of infection. Why do you need antibiotics? In genetic engineering, you need antibiotics. For example, ampicillin, tetracycline. I'm not talking about ampicillin resistant gene. I'm talking about antibiotics. Why do you need it? What are they going to do it? Do there, sorry, not do it. 4. I don't know what happened to my English. Yeah. So DNA fragments are what? Negatively charged, neutral. I think we did it previously, but still it makes more sense when we talk about it here, which is why I put it. So DNA fragments are negatively charged, neutral, either positively or negatively charged depending on their size. Positively charged. Such an easy question, came in NEET 2017, and still people get it wrong because we tend to forget the basics. I hope you would not be forgetting. Just imagine a DNA screaming around while passing through a lipid thing, and lipid, you know, it's hydrophobic, the inner core, so it has to be negative, which is why I told the answer, but still. Now, a gene whose expression helps to identify transformed cell is known as a gene whose expression helps to identify transformed cell. A gene which is normally expressing, and it is now helping to identify which one is transformed, which one is not. What do you call it? Vector, plasmid, structural gene, selectable marker. Again, 2017 question. So 2 questions, I just put it there. Both came from this chapter in the same year. So you can expect more number of questions. You get it? Okay, now, chalo, let's solve. So biolistics is suitable for, that is gene gun is suitable for what, children? For transformation of plant cells. Always remember, just blindly remember, for gene gun, plant cells. For microinjection, animal cells. For heat shock, bacteria. Works? Now for transformation, microparticles coated with DNA to be bombarded with gene gun are made up of what, children? I told you it's gold or tungsten which is wrapped around by the DNA and then it is being put. Okay. Now in genetic engineering, the antibiotics are used for, the antibiotics will be used as selectable markers, my children. Okay. Why do you use it? Selectable markers, because if there is a gene for this antibiotic which is being put there, a resistant gene, then obviously that particular organism is said to be transformed. Okay, and now I can mess it with the gene of interest also. So there are a lot of things for which I need a selectable marker, and your antibiotics helps in that. Now DNA fragments are negatively charged. There is no question regarding that. There is no positive, negative, neutral, nothing like that. It's just negative. Now a gene, and also please remember it's hydrophilic. Always people used to tend to forget this. Hydrophilic as in it loves water, just like I love you. Okay, so it loves water, children. A gene whose expression helps to identify transformed cell is known as, again it will be known as selectable marker. Its expression, if the selectable marker is not expressing itself or it is expressing itself, then you will get to understand whether it's transformed or not transformed. Okay, whether it is recombinant or non-recombinant, that is the thing. Okay, so basically this is the answer. Easy questions, no, but it becomes easy only when you study it. If you have not studied it and you're just there for attempting a lot of things, then it does not make any sense, right? Cool. Next thing, one little homework for you, my little cuties. In gene gun method used to introduce alien DNA into the host cells, microparticles of dashed metal are used. This came in NEET 2023. Can you imagine? Just 2 years back. Such an easy question again which is being repeated. Can you solve it out? Can you solve it out, my dear little cuties? You should be trying it out. Okay. So that's all. Now I am happy because I was able to explain the other thing properly, and I hope you are making utilization of this. Okay, every time when I go after teaching, I recollect what I taught. Is it right or wrong? There is another way of teaching it or not? And every time we teachers try to improvise ourselves when we get good students like you. So yes, definitely motivate us by subscribing to our channel and also putting down certain good comments. We'll love to see that. Okay, and probably I might not be replying to certain comments, but yes, I am definitely seeing it. I'm really happy, and I will definitely reply to all of that. And if you have any doubts, children, please be hesitant free. You have to ask the doubts. It's very important. You know, exams are around the corner, so please don't think that who will think. Basically, this is just social media, and there is no one who knows your identity. So basically, please feel very free. And even if someone knows your identity, you should not be ashamed of asking doubts, because doubts brings out inventions, discoveries, and a lot of things. So yes, that's all, and I hope you have a wonderful day, my dear children. Bye bye, take care.